I put announcements on the blackboard a bit more often than Dr. Chen does. I also email them to you, so hopefully you read them in advance. One of the things that I told you already last week in preparation for today is that all parts of this class build on the earlier parts of this class. So everything that you learned, I think I used the example, everything that you learned about the structure of membranes from Dr. Chen is going to be relevant now. Other things about the mitochondria, and I'm just going to pick up from where we left off and go into more detail about cells. And one of the things that's really very important as we became eukaryotic cells, the cells that make up a high proportion of our body, is that we developed what's called a revolutionary time. We developed what we call the endomembrane system. The endomembrane system is the basis of a great deal of Western medicine. So if you were trying to figure out why insulin isn't being secreted, insulin is secreted off of the endomembrane system. Prokaryotic cells can't do that. They don't have an endomembrane system. One of the major key differences between prokaryotes and eukaryotes was the evolution of the endomembrane system. And you may not know it if you go off to the NMD, they may not talk about it that way, but if you're not a surgeon, if you're almost in any other area of medicine, you're going to be involved with things in the endomembrane system. The medications that you take for blood pressure are fake ligands or triggering receptors to be synthesized. This is all coming off of the endomembrane system. If you're using cholesterol-lowering drugs, again, it's coming off of the endomembrane system. What we're going to spend the next 2 lectures on is talking in detail about the endomembrane system, but to have you to reinforce in your mind something that at least when I teach Bio 181 I include, but Bio 181 is much longer when I include this, is the endomembrane system. We're going to briefly cover it so that you understand the changes that occur, maybe in just 15 or so minutes with this slide set here in front of you. And then we're going to go on into what's in your bodies right now. There are two big changes, well, there's a whole bunch of changes that occur between prokaryotic cells and eukaryotic cells. One that I know that you've heard about from somewhere, even potentially from high school, and also in Bio 181, and I presume in 281 also, is it's called the theory of endosymbiosis, where one cell ate another cell. And we're going to talk about that and put it into context, but that is not the endomembrane system. I repeat, getting chloroplasts and mitochondria in your body, which I might still put on the test, but it's not part of the endomembrane system. Does anyone know in advance what the endomembrane system is? How about young lady right back there with that straw? Do you know what the endomembrane system is? Do you know what it is? What's it composed of? It's okay if you don't, but I wanted to hear it. What? Louder, please.
What are you asking about it?
Anything that you know about the endomembrane system, I'd like you to tell me now. It has what in it? Membranes in it? Yeah, that's good. And what else? And it has lipids, which could be a little bit redundant with membranes. Anything else come to mind about it? Protein synthesis could occur on it, yes. Anything else? No? Do you know what membrane flow is? Do you know what co-translational insertion is? Well, we're going to talk about that in the next 2 lectures. But it's good that you know that the membranes are involved in this system. So let's take a look at this first slide. I don't need to make it bigger. So, 2 major changes. We're going to talk about a few others at a very minimal level, but the 2 things the one thing that I want to draw your attention to is item number 8 of these internal membrane structures. And they're the internal membrane structures that were composed by the endomembrane system are, and you're going to write them down or type them down now, are the nuclear envelope. That's not to say the nucleus proper. The nucleus proper contains the DNA. But the nuclear envelope, the endoplasmic reticulum, and there's 2 parts to the endoplasmic reticulum. There's the rough, don't forget to write this down. There's the rough endoplasmic reticulum, which is closest to the nuclear envelope in general. And there's the smooth endoplasmic reticulum. which is branching off from the rough endoplasmic reticulum. Don't forget to write that down, it's going to be on the test. And those three membranes that I just told you, to repeat those membranes, the nuclear envelope of the nucleus, the rough endoplasmic reticulum, and the smooth endoplasmic reticulum are contiguous. Contiguous means there's a pathway that you could walk on. Just like you could walk, if we went outside this building beyond the construction level, and you were standing in front of the student union, you could find a sidewalk to walk on all the way over to the core building. There would be one continuous sidewalk because the slabs of the sidewalk are contiguous. The lipid bilayer in the membrane is contiguous. It means there's a constant flow of the two lipid layers from the nuclear envelope, the rough endoplasmic reticulum, to the smooth endoplasmic reticulum, and that's going to be important because that's going to be by backflow. By backflow, did you write that word down? Backflow? What? Oh, okay, that's different then. By backflow, it was one of the announcements I made was don't video record me because I don't want to see my fat stomach and my nose somewhere on a YouTube video. But if you want to audio record me, and even if you want to share these notes, it's not going to be the same thing as being here. Trust me, if you aren't here, you're going to get a C at best on the test. But I warned you now through the announcement page, and I warned you just moments ago verbally. You need to come to the lecture if you want to get more than a C in the class. What was the last thing I said about the lecture? Backflow. Why is that so important? Because that's how the nuclear envelope gets lipids. That's how the nuclear envelope gets proteins. Lipids are going to be synthesized in the endoplasmic reticulum. Some people would say specifically the smooth endoplasmic reticulum. And proteins are going to be synthesized in the rough endoplasmic reticulum. The lipids backflow into the rough endoplasmic reticulum, and then they backflow into the nuclear envelope, not the nucleus, the nuclear envelope. Proteins are synthesized in the rough endoplasmic reticulum and backflow into the nuclear envelope. And they forward flow into the smooth endoplasmic reticulum. And I'm going to ask, I'll take a question in a second, but I'm going to ask, how can they do that? But let's see if that's what your question was about.
I was going to clarify, do lipids backflow into the
Lipids are synthesized in the smooth endoplasmic reticulum and backflow into the rough endoplasmic reticulum and into the nuclear envelope. Proteins are synthesized in the rough endoplasmic reticulum and backflow into the nuclear envelope and forward flow into where? Smooth endoplasmic reticulum. These words, backflow and forward flow, would be what would be happening in a stream where the water is contiguous. If you were going forward flow, you would be going with the current downstream. If you were going backflow, you would be having to paddle to go against the current, to go upstream. Okay? How do lipids move that way? Dr. Chen told you. How about you with the pitchfork shirt on? How can lipids flow forward or backwards in the membrane? How about you? It's okay not to know, but I call this to your attention that you don't, so that you will know that this will be a test question. So, how do lipids flow forward or backwards in the membrane?
Potential.
Say again?
Potential.
Potential. They potentially could do that, but how do they do that? It's okay not to know. How about that person in the last row with the red shirt with your hands sort of like this? You don't want me to ask you? There is some truth to that, but you'd have to say several more words around hydrotropic interactions for me to know that you knew what that meant. Let me ask anybody this question. Who's going to answer it? Are the lipids covalently linked to each other? No. So, what's unique about membranes? They're fluid. The lipids, on one lipid layer, the lipids can move. It's called the fluid mosaic model of the membrane. The proteins are also fluid because the proteins in general, I can talk about graphs at this point, but I don't want to talk about graphs. The proteins are also fluid in the membrane. So if I'm pumping a whole bunch of lipids into the smooth endoplasmic reticulum, and the smooth endoplasmic reticulum is crowded, everybody was in this room and I started pushing a new lipid in, I would be shoving back flow and I would be shoving forward flow. Okay? Bummed you that I got this slide. I took the mouthwash here so I wouldn't have that breath. All right. So we're going to talk more about that system, but first we're going to talk about how it evolved. And then, because some of you confuse, I know that you confuse this because I teach this in the summer online, and I teach this almost every year, and I know that even though I tell you this, you don't believe me. And you answer this, you get a point off when I ask you, is endosymbiosis the same thing as the endomembrane system? And you say yes. Don't say that. Endosymbiosis is how we got chloroplasts to mitochondria. We'll briefly describe that, and when I do, you'll see it's very different than the evolution of the endomembrane system. But they have one thing in common. Both the endomembrane system and endosymbiosis required losing the cell wall. Repeat, required losing the cell wall in this ancient precursor to a prokaryotic cell. We have no idea, and I have to say a few very general few words about evolution. We have no idea that those things that, we have no strong evidence to indicate that those things that we call prokaryotic cells that exist in the world today are the same thing that existed 900 million years ago. There's a good chance that they evolved forward, but not the same exact species that was present 900 million years ago is present now. But we think they're present. Primitive. We think prokaryotes are primitive. And as human beings made out of eukaryotic cells, when we draw the tree, we call them prokaryotic cells and we say they're more primitive. When you want to try to distinguish between what used to exist and what exists now, I would call that type of ancient prokaryotic cell, I would call it the primitive. Proto is the precursor or prototype of primitive proto-prokaryotic cell. And the primitive proto-prokaryotic cell, which existed 900 million years ago, may not exist as a living species today. When a species ceases to exist, what's it called? What? Extinct. When a species ceases to exist, it's called extinct. When a species is still alive, it's called extant. E-X-T-A-N-T. So let's look at these signs. So, the primitive proto-prokaryotic cell had to go through a whole bunch of things to become the primitive proto-eukaryotic cell. Because those eukaryotic cells that existed 800 million years ago might not really be alive now either. In fact, when you take classes that deal with evolution, they'll point out that life may have started on this planet hundreds of times, and some rock slide may have destroyed it. Life ceased, and we had to wait another couple million years again, in addition, for life to occur again. But both of them, when they occurred, had to lose the cell wall, and I'll explain why in a minute. And the cell wall, you've heard me say, the cell wall led to endosymbiosis, led to this B is the endomembrane system. Okay? Now let's go on to the next slide. What good is a cell wall? I wish I could have found this slide. I would have showed you this, but you'll have to put up with my really bad art. This, hello? Oh, I have to press the button. This is a mountain. This is a mountain. This is a lake. That's flat land, and there's flat land on the side. This is full of water with solutes in it. Why is a cell wall useful? This is a rain cloud. It's a terrible looking rain cloud. This is rain. Rain is hitting the water. And it's a big 4-day thunderstorm. That's a lot around mountains sometimes. What happens to the solutes in the lake? What? They dilute. Now let's pretend here's a window diagram. Here's a cell, primitive proto-prokaryotic cell. This is in my way. A primitive proto-prokaryotic cell with its cell wall around it. When that cell ends up in this dilute water because of the rain, what's this cell going to try to do? That louder, I heard it, but I didn't see the lips move.
Match the concentration.
Something to concentration?
Match it.
Match it. Well, what if it rains some more? It's matched it, but now it rains more. What's it going to do to the solutes? It's going to dilute the solutes. When it dilutes the solutes, and there's a lot more solutes in the primitive proto-prokaryotic cell, what's the primitive proto-prokaryotic cell going to do? And it'd be gone. What keeps it from being gone? The cell wall. How many of you in your apartment somewhere or in your house have a washing machine? That tubing that connects the washing machine to the faucets that are filling it with water. Tell me what it's made of in your house. Didn't you ever have that? What's it made out of in your house? You know? Anybody look behind their washing machine? How about you engineers? Ever look behind your washing machine? It's not made out of plastic, is it? What's it made out of? Is it made out of black rubber? Who's is made out of black rubber? You have a time bomb in your house, right? Because as that ages, the rubber is going to corrode, and if you don't turn the water off at the valve, the pressure is going to cause it to rupture. Who has stainless steel mesh surrounding it? That's the way to do it. It costs more. You won't come home and find your kitchen flooded all the way into your living room. There. Life is everything that engineers have thought of. Life can do things better than engineers can think of. When I teach this whole class to myself in the summer, I actually have like 20 minutes I lecture on engineering feats of life. But having that metal mesh around your washing machine is protecting it from exploding. That's exactly what the cell wall is doing. The cell wants to go whoop, but instead of just being a plasma membrane out there, go whoop, and it reaches the cell wall, which is like a metal mesh. It's porous, but it won't let the plasma membrane explode until the dilution becomes even greater. You had to have the Earth's environment settle down to a much more stable state, that you didn't have fluctuations in large water masses where life could appear. And then, mutations could occur, and you have the loss, you have the loss, I can't get lost in here, loss of the cell wall. A very dangerous situation. And if it rained, you could die right then. Primitive proto-prokaryotic cell. And if it dried out, you could clean it, but you can clean it in both cases. That's osmosis. You learned that in Bio 281 and 181, and probably in Bio 150. So, there was never any purpose. But by the way, the reason there are these blank spots in the slide, these are my online Bio 53 classes. I teach that as an I course in the summer. So this is where I actually would be writing on the screen in front of the students, but I had to use this. But I had to use these two blank screens. This is where I write on these screens, but I had to use the paper a moment ago because I can't write the same way here. This is an extant eukaryotic cell. This extant eukaryotic cell is viewed by transmission electron microscopy over here, and in an artist diagram over here. And I really don't have time. I only teach half the course to talk about the limitations of technology, but they're very important to the limiting the advancement of science by not understanding what these limitations are. In this transmission electron micrograph, one of the things that you see is that there are no internal membrane structures. That's one major difference between prokaryotes and eukaryotes. No internal membrane structures. There's a plasma membrane, and everything else is floating or somewhat organized in an aqueous mass in the center of the cell. Some of you who have had a microbiology course might say, what about the mesosome? If you don't know what the mesosome is, don't worry, but for those that would invoke that, the mesosome is infolding of the plasma membrane, not an organelle, not an internal membrane. An infolding of the plasma membrane with the circular DNA is attached to it. If you don't know what the mesosome is, don't worry, it's not going to be on the test, but I want to settle the minds of microbiologists for a moment. If we look over here, we're going to walk our way in down to the cytoplasm. And not all primitive proto-prokaryotic cells, presumably, and certainly not those that are extant today, have a capsule. Some of them do. These are when they're gram-negative bacteria and things like that. And then we have the thing that confused eukaryotic biologists for a long time, because back before microbiologists understood the chemical structure of these things, they began using this terrible term right here, the outer membrane. And then high school teachers began to teach that the outer membrane of a bacterial cell is clearly the outer boundary of life. But the outer membrane of a bacterial cell is a cell wall. And the cell wall is not the outer boundary of life. It's always the plasma membrane. But because the high school teachers got it wrong, and you got it in your minds at a young age, the people who wrote SATs, GREs, and MCATs as a trick question, a really simple trick question years ago, began to ask, what's the outer boundary of life? And you can be sure they said cell wall. Sometimes they even said outer membrane. And if you check that, you were wrong. The outer boundary of life is always the plasma membrane. There is no nucleus, there's circular DNA that's supercoiled, and there are ribosomes floating around in the cytoplasm. Supercoiled DNA is this area here, and then there was the rest of the cytoplasm that had free-floating enzymes in it. The endomembrane system, with the exception of chloroplasts and mitochondria, the endomembrane system made all of this. The eukaryotic cell is so big you can only have little vignettes of the CEMs, but there's the nuclear envelope right there. There's the rough endoplasmic reticulum going out to the smooth endoplasmic reticulum, going to the Golgi apparatus, making vesicles. Just the mitochondria and chloroplast, where's the chloroplast? Well, it's an animal cell, there's no chloroplast. But just the mitochondria, and in a plant cell, mitochondria and chloroplasts are going to come from endosymbiosis. This happened because you lost the cell wall, and when you lost the cell wall, you allow the plasma membrane to have a function. There was never any use for the plasma membrane to become flexible in doing things before you lost the cell wall. But once you did, then you could make the plasma membrane invaginate into the cell. The next slide will show that. Before I go there, what do membranes do? Anyone know from Dr. Chen's lectures what membranes do? Well, good. Then I don't have to. Oh, go ahead, tell me. Protect the inner environment from the outer environment. That is true. That's one way to say it. Is there another way to say it? Yes, go ahead.
I was going to ask you something on the previous slide.
Go ahead, Jake. Then.
What did you mean by not the lack of physiological shape?
I couldn't hear what you said.
What did you mean on the last slide where it said note the lack of physiological shape?
Ignore that. That's for the online class. Those red notations that you see in these slides, that's where I had a commentary, and I lectured about that online, but that's when I was teaching the whole class and had more time to do that. Every time you see red text, yes, green shirt. What? You're selectively permeable is true. Go ahead and far back. Yell, please. I can't hear you, I'm sorry. I'm going deaf in my old age. Do you know I'm retiring in 3 years? So I'm an old man. You have to really speak up. Eh? What'd you say, Cindy? But let's go on. I wanted you to come up with some. You all said correct things. And if you are, there's bumpers and splitters. If you're a splitter and look in books, you can find 35 different things that membranes do. But for the remainder of the course, when I teach it, because it really is, I'm a bumper. Membranes do two things. They partition water, and it's the lipids in the membranes that partition water, not the protein. But they partition water because of the hydrophobic nature of the lipid bilayer in the center. And things that are soluble in water. So, yes, they separate the inside environment from the outside environment because the water that's on the inside of the cell doesn't easily leave to go to the outside of the cell, and all the enzymes that are on the inside of the cell, changing the environment of water, are uniquely there because they're water soluble and they can't move across the plasma membrane of the cell. You're partitioning molecules of life inside the cell from the molecules of death, or the molecules of non-life, not death, non-life, outside the cell. And the other thing they do is they compartmentalize enzymes to increase the efficiency of chemical reactions. And so when the membrane had a cell wall around it, it wasn't able to do anything with this outer surface. But you take a cell and you take the cell wall off. There's no cell wall. The outer boundary of life is just shown in the plasma membrane. And now the cell could make caverns into its cytoplasm just by using elements of the cytoskeleton and pulling the membrane in. And it pulled it in, and the circular DNA was sort of trapped here in the middle, and some of these things that came in came down there and laid around that initial primitive prokaryotic cells potential circular DNA and made the nuclear envelope around it. And these other structures became the endoplasmic reticulum, because all you have to do is seal off the plasma membrane right here, make these two areas fuse, and now you've got internal membranes forming. You've got the precursor of the endoplasmic reticulum and the Golgi apparatus, and you've got the precursor of the nuclear envelope around here. And this change required the loss of the cell wall. This change also required that the environment was not extremely variable, because you didn't want too much rain or too much desiccation occurring. And a reminder how difficult this would have been. And you know that the number of species in the tropical environments are much higher than the number of species in Arctic environments, for example. And that's what we had to have a period of time in a certain area of the Earth where there was less rain, less desert-like conditions. And once you stabilize some small portion of the environment, then you could begin to have the evolution of the primitive proto-eukaryotic cells. None of us were alive at that point in time, 1.2 billion years ago. But we can make progresses and we can use the fossil records once life existed to understand what was going on. But this is what's presumed to have happened. And this included then, what I've already indicated, the origin of a flexible cell surface after loss of the cell wall. Elements of the cytoskeleton came into the cell and began to move the membrane around in different ways, and also gave the cell a shape more complex than circular. The origins of the nuclear envelope, and they say digestive vesicles. I wish there was a primary lysosomes. I consider this primary lysosomes right here. And the list is going already. And my battery's too old. The endosymbiotic process that gave us mitochondria and mitochondrial chloroplast in plants also required the loss of the cell wall. But the people that deal with evolution, not just me, think that this was a separate evolutionary step to have endosymbiosis, and we'll briefly talk about it. But now you know what the endomembrane system is. You know that the endomembrane system, it's already in your notes, nuclear envelope, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the Golgi, the vesicles, and also primary lysosomes. Looks like the same side. And then, somewhere along, so here we have it again, just another summary slide for this. This is the primitive prokaryotic cell up here with its free-floating ribosomes on free-floating polyribosomes. It had a cell wall. It had circular DNA in blue. It lost the cell wall. The plasma membrane was able to have all these invaginations on it. The ribosomes are still in there. Some of these invaginations began to encircle the DNA, which ultimately led to the formation of the nuclear envelope around it, protecting the DNA even further from mutation by protecting it from some physical trauma that we haven't really talked about in this class. Vesicles began to appear. And what's on these vesicles? For those of you that sit closer, can you see these little dots on these vesicles? Can you see it right here? Here, here, here. Can you see those? Those are membrane-bound polyribosomes. That began a new population of proteins that were synthesized. That began a whole new system of protein synthesis inside of the cell. Prokaryotic cells always extant prokaryotic cells have free polyribosomes. Everything that you know from Dr. Chen for sure, you know that from him on how protein synthesis occurs up there. But you get another type of protein synthesis occurring when you have rough endoplasmic reticulum, and that's the precursor to the rough endoplasmic reticulum forming over millions of years. The nuclear envelope forms, and then elements of the cytoskeleton begin to push out on different parts of the cell. And right here, microtubules push out this really long thing in here, and this really long thing became the flagella. And a flagella is a wonderful thing. A flagella lets you move. Cilia, which are a smaller set of flagella in a sense, can also let you move. And if I was really hungry, and sitting over, see that guy over there in the far corner with the great bluish-gray shirt on? If he had a Big Mac and I was starving to death, but I couldn't move, he had that Big Mac, all greasy and everything, sitting on top of his laptop, just oozing into it, with the smell of onions and the secret sauce. And I really don't eat Big Macs, but you guys know what that is. And it's just sitting there, and I can't move. eat it. And I starve to death. But if I have a flagella, I can swim up the concentration gradient of the aroma and eat it. Or if you happen to have a hacksaw, Halloween's coming. What's that guy with a chainsaw that cuts people's heads off or something? I don't watch movies, but they make lots of series about it. If that guy with the chainsaw was coming at me, and I couldn't move, I'd be dead. But if I could move, I'd be out the door. You might kill somebody else, but you ain't going to kill me. So motility is really a handy thing to have. Plants don't have it. Plants have a harder problem when things like that happen to them. You ever see your rose bush run away from you when you come out with your slippers? I haven't seen that yet. All right. So that flagella allows motility. And motility lets you move into a better environment. Doesn't need to be a Big Mac. It could be where the osmolarity is better for you. It can be where the other nutrients that you need happen to be, where there's less potential for desiccation to occur. It's a wonderful thing being able to walk. I'm glad I'm an animal, not a plant. I mean, think of those four saguaro cactus. Those drunken people with shotguns out there. I always love it when a hunter is so stupid. He stands right under one of the arms of the saguaro cactus and is trying to break it off, and the thing comes down and crushes it. I think the guy deserves it. All right. Don't worry about that. Let's see. We'll go to here. Now, so everything up to here, ignoring this slide right here, this slide is going into symbiosis. Everything up to this slide that I talked about was the evolution of the endomembrane system. And then when I finish this first slide set, we're going to go on to more details about the endomembrane system. We won't really come back to very much to endosymbiosis. But endosymbiosis is where one cell eats another cell, and it helps the cell. It increases the fitness of it. This is the endosymbiotic theory. So endo for endocytosis, eating. And symbiosis because you came into a symbiotic existence. All right. So how does that occur? Well, it's more likely, the probability that it's going to occur more frequently is greater if one of the cells can move. One of the cells can move and the other cell is potential food for it, but it can't move. It's more likely to be eaten. And notice this red line is, Jake, this is worth noting. If this were the online class, you see me put a big X through the peroxisome. Your high school teachers and other people will tell you that the peroxisome was also involved by endosymbiosis. It involved by the endomembrane system. It was a mistake. It's been corrected. The textbooks haven't been corrected. You should put an X through this if you have a hard copy in front of you. But for mitochondria, the ATP powerhouse of the cell was created by endosymbiosis. So was the chloroplast. The chloroplast also had to have mitochondria in them. Even though it's the right here, for example. You can't eat a cell that's on the outside of you if you have a cell wall, right? If my fingers represented a cell wall right here, could I eat that cyanobacter that's right on your left side of my hand? No, I couldn't. So you still had, for the endosymbiotic approach or theory, you had to lose the cell wall. And this cell eats it. And this happened all the time. It happened for millions of years. But one day, this primitive prokaryotic cell that could synthesize, had some photosynthetic ability before this, wasn't feeling very well. It had the stomach flu. You ever had stomach flu? Remember, you're all biologists when I ask this, don't gross out too much. So what happens when you look down at the toilet after you get out and have the stomach flu? What do you see in the toilet? A brown mixture that has, if you were able to eat something earlier, a brown mixture that has food in it. Now, it's perfectly normal, by the way, it is perfectly normal to see corn in your stool, whether it's diarrhea or not. Corn isn't easily digestible. But you should never see tomatoes in your stool. You should never see peas in your stool. They're very easily digestible. But when you have the stomach flu, your digestive tract doesn't work particularly well. Well, this big fat cell right here had the flu. And it ate this cell right here that was outside of it, and it surrounded it with its own plasma membrane with one membrane layer, and the plasma membrane of the cell, so you had two membrane layers in there. And this was cyanobacter, which is a extant version of something that was prokaryotic and could photosynthesize. And there it sat. And what did that do? You know photosynthesis from Dr. Chen, don't you? What are the end products of photosynthesis?
Glucose and oxygen.
Glucose. What does glycolysis require? Glucose. When glycolysis uses glucose oxidatively, what's the net gain of ATP? I have a root 36, 38 gross. I was rushing along, sorry. So here's this cell. It's sick. Can't digest its food. It's not getting enough ATP. It's getting sicker and sicker. Somewhat. This is the equivalent of chicken broth with a little bit of kanji, rice, in it. And it gave the cell enough strength to survive. And that cell survived and became healthy. And this cell still made, and this cytosine cell still made glucose. Made energy. Made this cell a supercell. It was a really happy cell. And it decided, since it made it through this trauma, this has more of speaking decided, it increased the fitness and kept those chloroplasts. And these were the cells that became our conifer trees and our grasses. And anything that's eukaryotic that can photosynthesize. Same story would go for mitochondria. The ATP story still holds there. Except now, this is cell could really make lots of ATP, and this was the 6 cell couldn't make ATP and ate it. Made lots of ATP and have another supercell. That's how it came about. But, and it is not part of the endomembrane system. Do you see the endomembrane system is already there? So if I ask you, did mitochondria and chloroplasts come from the endomembrane system? Are you going to say yes? No. Okay. So now you know. Now I can go on. Let's just see if there's anything else on these slides. That sort of says everything I said. The only thing about I will add one thing about peroxisome, just I have a hard time realizing this. I mean, you don't know how many. If you go back and you look through the fossil record, you're talking to a paleontologist, they will tell you how many times life potentially started on this planet and was crushed by a landslide or crushed by a comet hitting us or something. I think things started many, many times. It's a miracle that we're on this planet. Okay? And our average lifetime to be here is about 10 million years. Our average lifetime is at least something less than 1,000. Anyways, it's small. We're not going to be here forever, and we're doing a great job with global warming right now, right? So we're going to be here an even shorter amount of time. But back in the ancient Earth, if I could transport you, or me, but I wouldn't want to be there, back on the ancient Earth, you know what happened in 3 minutes? I'd drop dead. Because there's no oxygen on the ancient Earth. All these organisms that existed 900, 800, 700 million years ago, all these ancient organisms were anaerobic. But look, how many ATP can you produce from anaerobic metabolism? It means you can't use any DH2 or FADH2 in the electron transport system. Well, you're not going to have any. And I wouldn't survive. Oxygen was actually toxic. And the peroxisomes, which came not from endosymbiosis like your high school teacher would have told you, but from the endomembrane system, the peroxisome's job was to get rid of the toxic oxygen. We actually evolved a mechanism to get rid of the microscopic amounts or trace amounts of oxygen that were around. And this is just, so Earth supposedly formed 4.5 billion years ago, and it wasn't really in a place where life could exist at all. It was too hot, all these sorts of things. It became stable about 4.2 million years ago, and life began. So if the planet came into existence 4.5 million years ago, minus 3.5 million years ago, life began. We haven't been around as life in general on this planet for very long, and at the rate we're going, it won't be very long. But I'll be dead because I'm retired. You'll And if you want to look at those slides again, there's just another set of slides you can look at. On your left is the endomembrane system, and your right is endosymbiosis. We're going to go on to, now what I've just mentioned I will be on the test. Don't turn, you're not, oh, that was the introduction. I don't need to remember this. No, I will ask 5 or 6 questions about everything I just taught you up until now. So be sure to keep track of what I said today. I also want to tell you that the way I teach the class is I try to teach it in layers, which like the concept of the onion. I try to take one pill off the onion and then the next pill off the onion and tell you more and more detailed things as time goes on. So I started at a very general level, something not too complex, although you aren't that familiar with the endomembrane system. Now you're more familiar. And I'm not going to speed up, but I'm having you get used to the way I talk. And now begin, as we go into these next things, I'll begin giving some examples. Some people, when I was young, I'd do the same thing. I would give all 35 examples that you should know if you're a graduate student, but you're not a graduate student. So I don't want you to know that level, but I want you to know a model of how something would work, and then when you learn of a new protein that can function in that way, you can just add that protein to your knowledge. I'm not going to tell you everything all at once. I'm just going to slowly, progressively go through. Everything is going to tie into this through the rest of the course almost. Maybe not in mitosis, but everywhere else. Everything is going to tie into the endomembrane system. That's how central it was to eukaryotic cells. So let's go to the next slide, 7. Well, actually, pause. Is there any questions? Are there any questions? I didn't blow anyone away, right? All right, I'm going to go on. So we're going to close this. Start that's smaller. Minimize this. Down here, I think I did. So now we've got membrane flow and co-translational insertion. Membrane flow and co-translational insertion are all about the endomembrane system. What co-translational insertion means is, what's the word translation mean? Yes?
Turning DNA information into protein through translating mRNA.
Well,
From one language to another.
I do know one language to another, but you jump too many languages. Because transcription. So let's start here for a second, just to have you see this. If you were a court scribe or a medical scribe, and you're there with the MD who's paying you, or the coroner who's paying you, what are you doing? Are you translating what he says, or are you transcribing what he says? You're transcribing. Right? Sorry, I said he, I said she. You're transcribing what that person said. Transcribing doesn't change languages. DNA is in the language of nucleotides, and RNA is in the language of nucleotides. That's why it's transcription. Translation goes from the language of nucleotides to the language of amino acids. That's going from French to English. That's called translation. Co-translational insertion, then, is unique to the endomembrane system, and it means that you have to have, for protein synthesis to continue, you have to be attached to the endoplasmic reticulum. Protein synthesis will not continue if you can't attach to the endoplasmic reticulum. It's co with protein synthesis in the endoplasmic reticulum. Insertion, the word insertion is used in a lot of sewing machine analogies, actually, historically, used for this event because it behaves a little bit like an electric sewing machine that has spools and bobbins, and the needle goes down and the thread comes through it. You'll hear on some of the movies I'm going to show you the sound of the sewing machine. I didn't put it there. That's the way it was originally described, because it seemed very much like that historically. The scientists were doing that. All right. So,
What is the acid attached to?
start again.
Yeah. So you said protein synthesis to occur to go forward, something that's attached to the ER. What is
The polyribosome.
this?
Polyribosome, which is different than a ribosome. Polyribosome is a messenger RNA and multiple ribosomes. The polyribosome has to be attached to the plasmid. Okay? And it's going to insert through a pore that we're going to see in this lecture. All right? Water. Let's see what the first slide says. The first slide reminds you that there's a handout. which is in the PowerPoint right after. So, not that you need to grab it now, but if I don't finish the lecture today, you ought to bring it for tomorrow. It looks like this. It's 2 pages long. The most complex part of this is what I'm about to do next. And I've written it out for you here. There's a few spelling errors and stuff. But I know it's really hard to grasp all this and write it all down. So I've written it out in advance. It's this page long. Let's make it smaller so you can see. It's this page long to here. From here to here, we've got a protein in the rough endoplasmic reticulum. But you've got it written in front of you. If you have it in front of you now, you can just correct words and a few sentence structures that I didn't write properly. I will ask you several questions about this. You know that 5 lectures from today, we'll have an exam. So I really only have 4 lectures with you. And look how much time I'm spending on the endomembrane system. And look at this. This is the only handout I think you're going to get in that time period. If you haven't noticed by now, you haven't. But when you get a handout, it's going to be on the desk. You need to know this handout, but it's right there in front of you. Just annotate it in some way. And I sent you an email about this morning. Okay? All right. So let's go to the next one. There you can see what I've just told you. And you can see how it links. Lecture 4 refers to my online class. It tells you that you can look up some things in the book if you need it. But I'm going to lecture on all that now, so you don't really need it. What you know about for sure from back in high school times is the synthesis of proteins, which is translation, from a 3-polyribosome. This pathway does not make transmembrane proteins, nor does this pathway from 3-polyribosomes make ligands. They're going to be exocytosed from the cell. It's the membrane-bound polyribosomes that evolved with the endomembrane system to create the internal membranes that lets you make receptors and ligands. Receptors and ligands, the types that we have, are very unique characteristics of eukaryotic cells, made possible by the endomembrane system. And then this one, when you have a messenger RNA, and there's only one ribosome associated with it, you know what you call that? This is not a joke.
A monoribosome?
A monoribosome. A monosome is vapsine, and a monoribosome, which is what you said, is the correct answer. Whoever said that. Okay. But once you have 2 or 3 or 4 in one, begin calling it a polyribosome. Certainly by the time you get up to 7, you can certainly call it a polyribosome. Okay? The ribosome always binds at which end of the messenger RNA? The 5' end. And in a sense, you can view that as the front end of the messenger RNA. The ribosome always binds there. And where does it move to? Where does the ribosome move to? Again, please? Where is the 3' end, which could be the back end of the messenger RNA? The front end of the mRNA makes the front end of the protein. The back end of the messenger RNA makes the back end of the protein, and if I wanted to, I could always go back to DNA polymerase 3, which makes front ends and back ends all the way to there. It's not what DNA polymerase 3 does, but it's already covered. So this is the cell. I'm just going to briefly, so you can see this just a little bit better, make the room a little bit darker. Then I'll turn the lights back on. The cytoplasm in a eukaryotic cell, the cells that make up a lot of the cells in your body, are really packed with things. This is a thin section electron micrograph, so it's a thin 2-dimensional slice, like a slice of salami or a slice of pepperoni that you'd use in that long stick of a Genoa salami or a stick of pepperoni. It's packed. The rough endoplasmic reticulum is shown over here with ribosomes attached to it. In some other area it becomes a little bit more tubular, which is the smooth endoplasmic reticulum. The nuclear envelope is right there, all those parts of the endomembrane system. These are mitochondria, there's no chloroplasts, this is an animal cell, but it's packed. And 3 polyribosomes do everything that they did. Lights are coming back on. 3 polyribosomes do everything that they did and still do in prokaryotic cells. Membrane-bound polyribosomes create those unique features that make you eukaryotic. Standard prokaryotic cells do not have membrane-bound polyribosomes. That's the membrane system again. I think I've talked enough about that. But I do want to show you something I've said. So the first element in the endomembrane system that I've given you in that list is the nuclear envelope. Not the nucleus, the nuclear envelope. And right here you can see it being, where is it? Right here, right up there, you can see in this diagram it's contiguous with the rough endoplasmic reticulum. And somewhere right over here, here's another place. There's a nuclear envelope contiguous with the rough endoplasmic reticulum, and then contiguous with the ribosome-free area of the endoplasmic reticulum that we can call the smooth endoplasmic reticulum. That has what type of transport you already know from my lecture earlier today. It gets new lipids and proteins because it can backflow, it can forward flow because it is contiguous. And the fluid nature of the lipid bilayer allows that lateral flow. But when it allows that lateral flow, let me just stop and see if these guys have questions. You want to ask me that question? I'm happy to answer it if I didn't see you put up your hand. You look like you were checking something in your notes.
What was the word? Is it contiguous or contiguous?
Contiguous. C-O-N, right? Contiguous. C-O-N-T-I-G-I-O-U-S. Repeating. C-O-N-T-I-G-I-O-U-S. Not contagious, by the way. All right? That movement can occur because you have lateral flow of lipids in the individual lipid layer, the lateral flow of proteins across that. Can a protein, if I had a membrane right here, if this were a membrane, a lipid bilayer right here, here's a transmembrane protein. This is outside, my fingers are outside the cell, my elbow's inside the cell. Could it do this? Oh, no. Why not? Thermodynamically unfavorable because we're putting charges through the hydrophobic area, right? Could a lipid go from this lipid layer to this lipid layer? One with an enzyme. Can't do it by itself. The flipase could do it. Scramblase could do it, but not by itself. Never happened, but it can do this. It can do this. That's forward flow. Picking a direction. That's back flow. Okay? I'm getting too old to lay on the floor. All right. This is just for your identification. Some people wonder how many mitochondria are in a cell and stuff, so I don't have to tell you later. I just put it here in the book. Tell you a few more facts. This previous slide and this slide won't be on the test. It's just here for you to orient yourself if you want to. These sequences won't be on the test, but this is really important. Briefly, just so you can see the number of amino acids in some of these things, let me dim the room, which you won't need to come with these specific amino acids. This is the first one we're going to talk about when we get to it. It's amino terminal end starts with methionine, as you know AUG is the start codon. And it'll go for quite a long distance, and then you'll see retention in the lumen of the ER. These are signal sequences. These are really important. Come back and visit again. Bye-bye. So long. I guess they don't like transporting the membrane. Anyways, so you're going to find imported to the mitochondria, imported to the nucleus, exported from the nucleus, imported to the peroxisome. These are all tickets in a sense. Tickets just like you'd use if lights are coming back on in 3, 2, 1. These are like going to the airport and paying a travel agent to buy you a bus ticket to get to the Los Angeles airport. At the Los Angeles airport, you get on a little tram to get to the right terminal. You have your ticket that says, take me to Frankfurt, Germany. You fly to Frankfurt, Germany. You have a train ticket to take you to Dusseldorf. And then you get on another bus in Dusseldorf and go a little bit further. Each one of those tickets you have to use are the same effective ticket that you have to use to get a protein and a lipid, or proteins out of its membranes, to different parts of the cell. There's a different ticket for everywhere you're going to go. So you can imagine peroxisome being Dusseldorf. You can imagine the Los Angeles airport being rough endoplasmic reticulum. The one sequence that I'm going to ask you to know one general thing about is this first sequence up here, this first ticket, and I'll show you how it's used in just a minute. This first sequence is, in general, more than is shown here, write this down, because you will have to notice on the test, is 15 to 60 hydrophobic amino acids. Not all of them are hydrophobic, most of them are 15 to 60 are hydrophobic. That's going to have an effect on where this protein is going to go. If you think a stretch of protein that likes to be, bye-bye, come back and visit again. If you think that a stretch of protein, I got 20 minutes in class. It's not just like she's probably not going to pass. So, wouldn't you like to know if I told you you weren't going to pass? You'd start writing letters of complaint right then, wouldn't you? You're predicting my life. What do you think you are, a fortune teller? No, after 32 years. In fact, this coming January the 2nd, I'll taught here at ASU 33 years. I know. I've talked longer than you've been alive. I know if you want to leave 20 minutes early and you don't have diarrhea, and you don't have some other medical problem, then you're probably not going to pass if you consistently do that. So you should come to all my lectures, and you should get an A, and you should not come late or leave early. I'll tell you a few times, and then it's all up to you, but I'm always calling it for your attention a few times. Yes, sir?
So you said for the 15 to 60 hydrophobic amino acids, is that just for the ER or is that for all of them?
That's the very first one, the one that says, take me to the rough endoplasmic reticulum. That sequence actually has a specific proper noun name. That sequence, proper noun name, which is the one I'll use on the test, actually has two of them. The one that's more typical, it's called the signal sequence. The signal sequence, S-I-G-N-A-L, S-E-Q-U-E-N-C-E. The signal sequence is the sequence that says, arrest protein synthesis and take me to the endoplasmic reticulum. It's also called the leader sequence. It's called the leader sequence historically after the name of the person who identified it. His name was Phil Leader. I cannot remember whether he was whether he was a professor at MIT or Harvard. He was one or the other. But people thought, the editors of the journal thought it was really funny that Phil Leader found the sequence that says, take me to the rough endoplasmic reticulum, like he's leading the rest of the messenger RNA there. As a joke, they put it in the manuscript. They called it the signal sequence, which we're calling the leader sequence in honor of the person who discovered this, and they thought the editor was going to make them take it out. The editor of the journal Cell thought it was so funny, he left it in. And so on MCATs and GREs, it is called both, that one that says take me to the rough ER, is both called the signal sequence and the leader sequence. Remind you again that you have a handout. And I'm going to take a moment to show you that out here, these little dots that you see are ribosomes on the rough endoplasmic reticulum. The rough endoplasmic reticulum is a sort of flattened area. For those of you that don't sleep in a sleeping bag all the time, although I could use a sleeping bag analogy, if you had a fitted bed sheet, and you have a fitted bed sheet laying here like this, and you put the sheet that you're going to lay between here like that space where you lay in bed is called the lumen of the rough endoplasmic reticulum. When something is in the lumen of the rough endoplasmic reticulum, it's a free-floating protein. Write that down. When something is in the lumen of the rough endoplasmic reticulum, it's a free-floating protein. Something that's in, say one more characteristic of it. When something's in the lumen of the rough endoplasmic reticulum, there's a high probability that it's a ligand. It's going to be exocytosed from the cell is going to be its ultimate fate. And the sheets are the lipid bilayers that make up the walls. And if you're a transmembrane protein in these sheets, write this down, you're in all likelihood going to be a receptor. And ribosomes are actually adhering to the surface of the rough endoplasmic reticulum. Why? And the answer is not clearly shown in pictures in your book. Because there's enough. Scientists write books. They didn't give it to somebody who's an artist, ask the artist to draw it. The artist begins drawing all these things, and it becomes very messy and noisy. And then they leave things out to make it simple for you. In this edition of the book, not in previous editions, but in this edition of the book, they left out what holds the ribosomes. What specifically holds the ribosomes on, and you'll be able to remember this name pretty easily, but it's not as shown as a picture in your book, is called the ribosome receptor. The ribosome receptor is a transmembrane protein in the rough endoplasmic reticulum. Why the rough endoplasmic reticulum is rough is why it has ribosomes. And it grabs onto the ribosome. We're about to see that. This is the book's diagram. It's left out the right. This book diagram is actually showing you 3 polyribosomes on the top, membrane-bound polyribosomes on the bottom, and down here it's left out the ribosome receptor because it was one more transmembrane protein, and they decided, the artist decided, and the scientist, either the reader didn't catch it, decided, yeah, leave it as, it makes it too noisy. These are totally different populations in a eukaryotic cell. Like, we start. Which one of these is present only in a This is a eukaryotic cell, and this is also a eukaryotic cell. But prokaryotic cells only have this type of protein synthesis. Some people have to get funny and say something on the way out. Sometimes it's not polite. These codes are totally what the population of messenger RNAs that code have no sequence that says take me to the rough endoplasmic reticulum on the protein. Totally different proteins that are present are the ones that go to the membrane-bound polyribosomes in the rough endoplasmic reticulum. They don't change. They don't. One day you don't make an mRNA leave the nucleus, and it's working on a free polyribosome, and the next day it's working on a membrane-bound polyribosome. This is something that was decided at revolutionary time millions and millions of years ago. The proteins made from this co-evolved with what made you have eukaryotic cells. You still have to have all the enzymes of glycolysis. You still have all the other metabolic pathways to make melatonin and all those other things. But those things that are unique to eukaryotic cells that don't exist in prokaryotic cells, almost all of them, not all of them, but almost all of them are made off of this pathway. And medicine uses its injections of things and pills that you take to act on this system, put you back in balance, whether it's blood pressure, cholesterol, or a host of other things. What's going to happen? Maybe I should show you a movie, just to make sure you have this in your mind. Give me a second. Remember where I put this movie. I guess it's going to give me 30 seconds. All right, it must be down here somewhere. It's not here now. Yes. Okay. This is from my online class. Make it bigger. So, what am I going to do? I'm going to turn the lights down first. So you can see a little better. Okay. I'll start it and make it bigger. Why?
As soon as a messenger RNA molecule is transported from the nucleus to the cytoplasm, ribosomes begin to translate the sequence into amino acids. Typically, many ribosomes translate the mRNA simultaneously.
Do you see that there's a large ribosomal subunit here, a small ribosomal subunit there as they come on, the small binds first and the large binds second? That happens in both types of protein synthesis for free and membrane-bound polyribosomes.
Ribosome begins at the 5' end of the mRNA and progresses steadily towards the 3' end. New ribosomes attach to the 5' end at the same rate as the previous ones move out of the way. These multiple initiations allow the cell to make much more protein from a single message than if one ribosome had to complete the task before another could begin. When a ribosome reaches a stop codon, the ribosome and the new protein dissociate from each other and from the mRNA. This electron micrograph depicts a membrane-bound polyribosome from a eukaryotic cell.
That exists in your bodies and also in prokaryotic cells. And then we'll show you membrane-bound ones, and then we'll talk more about them afterwards. You're going to ask me someday if I can give you these, and I can't because they're part of the online class, and they're only copyright protected through the online class, so I can show them to you because I was watching. Bye-bye. Because I was watching them myself. But I can't let you have access to these, otherwise I'd get in trouble.
The endoplasmic reticulum, or ER, is the most extensive membrane system in eukaryotic cells. Proteins transported to the Golgi apparatus, endosomes, lysosomes, and the cell surface all must first enter the ER from the cytosol. As an mRNA molecule is translated into a protein, many ribosomes bind to it, forming a polyribosome. There are two separate populations of polyribosomes in the cytosol that share the same pool of ribosomal subunits. Free ribosomes are unattached to any membrane. Membrane-bound ribosomes become riveted to the ER membrane and translate proteins that are translocated into the ER. These membrane-bound ribosomes coat the surface of the ER, creating regions called rough endoplasmic reticulum. Two kinds of proteins are moved from the cytosol to the ER. Water-soluble proteins completely cross the ER membrane and are released into the lumen, while transmembrane proteins only partially cross the ER and become embedded in the membrane. All these proteins are directed to the ER by a signal sequence of small hydrophobic amino acids. The signal sequence is guided to the ER membrane with a signal recognition particle, or SRP, which binds the ER signal sequence in the new protein as it emerges from the ribosome. Protein synthesis then slows down until the SRP ribosome complex binds to an SRP receptor in the ER membrane. The SRP is then released, passing the ribosome to a protein translocation channel in the ER membrane. Thus, the SRP and SRP receptor function as molecular matchmakers, connecting ribosomes that are synthesizing proteins containing ER signal sequences to available ER translocation channels. In addition to directing proteins to the ER, the signal sequence functions to open the translocation channel. The signal peptide remains bound to the channel, while the rest of the protein chain is threaded through the membrane as a large loop. Once the protein has passed through the membrane, it is released into the ER lumen, after the signal sequence has been cleaved off by a signal peptidase located on the luminal side of the ER membrane. The signal peptide is then released from the translocation channel into the membrane and rapidly degraded. It is thought that a protein serving as a plug then binds from the ER lumen to close the inactive channel. But not all proteins that enter the ER are released into the ER lumen. Some remain embedded in the ER membrane as transmembrane proteins. For clarity's sake, the membrane-bound ribosome will be omitted to illustrate the translocation of transmembrane proteins into the ER membrane. In the simplest case, that of a transmembrane protein with a single membrane-spanning segment, the N-terminal signal sequence initiates translocation, just as for a soluble protein. But the transfer process is halted by an additional sequence of hydrophobic amino acids, a stop transfer sequence, further in the polypeptide chain. The stop transfer sequence is released laterally from the translocation channel and drifts into the plane of the lipid bilayer, where it forms a membrane-spanning segment that anchors the protein in the membrane. As a result, the translocated protein ends up as a transmembrane protein, inserted in the membrane with a defined orientation. In some transmembrane proteins, an internal signal sequence is used to start the protein transfer, which continues until a stop transfer sequence is reached. The two hydrophobic sequences are then released into the bilayer, where they remain anchored. In complex multipass proteins, in which many hydrophobic regions span the bilayer, additional pairs of stop and start sequences come into play. One sequence reinitiates translocation further down the polypeptide chain, and the other stops translocation and causes polypeptide release, and so on, for subsequent starts and stops. Thus, multipass membrane proteins are stitched into the lipid bilayer as they are being synthesized, by a mechanism resembling a sewing machine.
Lights are coming back on in 3, 2, 1. So we have 7 minutes or so to go. Let me close this. This one. Minimize this. So, bigger. So here is the small ribosomal subunit, the large ribosomal subunit is bound to it. The 5' end of the messenger RNA is not being shown. There's a protein coming out. It's being shown. This red area is the leader sequence composed of how many amino acids? 15 to 60 hydrophobic amino acids. And it continue coming out, except this thing called the signal recognition particle, the SRP, comes over and recognizes this. It's going to bind to it right here. And when it binds, it flips this back end of the protein right up to the place where new amino acids. Why protein synthesis stops is new amino acids new transfer RNAs, I should say, can't come in. So protein synthesis rests until you get rid of this brown SRP, signal recognition particle. Okay? For reasons I'll explain to you at the beginning of the next class, which has to do with the way biochemists did these early experiments, this was thought to be a rate-limiting step. It may still be a rate-limiting step. This is supposedly now, this unit, as a monosome, is diffusing around randomly in the cytoplasm until, by random chance, it bounces into this rough endoplasmic reticulum and finds the SRP, what? The SRP receptor. The SRP receptor binds to this unit, and a whole bunch of things happen. And the movie left out the ribosome receptor. And this still left out the ribosome receptor here too, but again, here's the SRP. Here's the 15 to 60 hydrophobic amino acids. The mRNA is shown on this one. The mRNA begins to move a little bit. The first 15 to 60 hydrophobic amino acids come out. This grabs there, plugs the hole where new transfer RNAs come through. This diffuses around randomly till it finds this thing. And that is the correct order up till there. At this point, it finds this thing. This thing is the SRP receptor. That's the SRP. I'm going to say this probably 1,000 times during the remainder of the semester. When the SRP is the ligand in this case, the SRP receptor is a receptor. Whenever a ligand binds to a receptor, there's a conformational change. That conformational change causes the SRP receptor to disassociate from, the 15 to 60 hydrophobic amino acids. And then, what's not shown is there's a ribosome receptor sitting right here that grabs onto the ribosomal subunit and holds the ribosome in place. And you recycle the SRP receptor. The movie did show that. There's the SRP receptor recycled right there. Some views of this process view the protein translocator channel as not plugged, but actually two halves, and they have to come together in space. That's the way this slide is showing it from Albert's other book, the Bible of Cell Biology, which is better than the one we use. But anyways, these two things come together. This and this comes together to make this channel right here. The hydrophobic structure of amino acids stay here. The ribosome receptor is still holding it. It's just not shown. And protein synthesis continues, pumping the newly synthesized protein into, at least temporarily, into the lumen of the endoplasmic reticulum. What's in an answer? There's a question. So, let's look at the next slide. Ribosomes are never embedded, they're attached. So if I had a lake and I had the boat tethered to the dock, the boat is the ribosome, the dock is the membrane. The boat can only go so far away because the rope is tethering. You can't move very far. What's tethering is the ribosome receptor, which is not shown. That's why this is, I think it's stupid and I put it in here. The ribosome receptor is there. And if I were drawing it then, if I was going to, well, actually I can't draw it in 25 seconds, but I will probably begin next class with the drawing. That way I'll prepare it ahead of time. You might want to take a picture of it with your cell phone or something, and then you can share it with each other. But I'll draw this process up here, and then